What group, what atomic group is in these heads? Look at the word. Phospholipid. Phosphate. If you get it through your head, now that phosphates have a negative charge, this next chapter is going to be so easy. Because we're doing biochemistry. Alright, let's see. Alright, I have to go back here. Alright, so your test has 20-something multiple choice. I think it's 24 multiple choice. It has 40 questions in all. The last 16 questions are answer in complete sentences and no more than two sentences. Answer this question. And they are math problems. You will have to calculate surface area and volume, and you will have to do surface area to volume ratios, just like on your math worksheet. You will also, and hey, the key to this is get the right units. And the formulas for calculating those are on your test. However, the formula for calculating rate and percent loss or gain is not on your test. Because I told you, the AP exam thinks you ought to know how to do that by the time you get out of 6th grade, and you should. So, here we have total water loss in milliliters of time in minutes. At 10 minutes, what was the water loss rate? At 10 minutes, what was the water loss rate?
What does it start out at?
What?
What?
Yes, it starts out at 0. You must use your calculator. At 10 minutes, what was the water loss rate? Kirsten! Can you tell me what numbers am I going to use?
Final.
What? I can't hear you. What? Why not? Because what? Who knows the formula for calculating rate? Oh, look at all these hands. Yes, ma'am. Tell me.
Rate equals any number over time.
There you go. Any number over time. What numbers is going to be our any number? 2.4 divided by 10. And our answer, the units for our answer will be milliliters per
Minute.
Milliliters per minute, because we're giving our time in minutes. Are there any questions about how to calculate rate? Alrighty, look at me. Drew Barry, oh.
Stephen Bell.
Y'all looking at all my boyfriend pictures.
Let's look at him. I bet he's hot.
No, that's actually scientists giving tops on stuff.
I'm just kidding.
Hi. If y'all want to see some cool videos,
I do.
TED.com, TED.com. Click on the biology. There's some cool videos there. It's mostly for AP, but there's, I mean, y'all will enjoy them, and they're all people that are talking about cool stuff. The other place, if you want to see, I don't know if you've ever seen this, and PBS.com. Go to PBS.com and search for Inside Giants, and they actually dissect things like elephants and boa constrictors, and I've downloaded them too. All right, here we go. See ya. Alrighty, listen at me. I need the percent loss or gain, percent change in mass for egg A after 2 days in vinegar. Don't look at your worksheet, actually work it out. Yes, you will need calculators for the test.
So does that mean?
What was the question?
Percent log, percent change. Hey, woohoo! Can you wait just about 2 minutes? Because I'm going to talk to you about your free response. Can you wait about 2 minutes? Percent change in mass for egg A after 2 days in vinegar. Yes, ma'am. Yes, ma'am. It is the final mass, 75.6, minus the initial mass, 54.6, divided by, oh, you gotta put your grams.
It's over there.
You don't put your grams, divided by your initial 54.6 grams. The grams cancel, equals times 100. There you go. I wish some of you would come up with a cute little saying for that. Like friends take away ice cream and divide by friends. I don't know. Come up with something.
Equals 0 ice cream. Your ice cream. 0 ice cream.
All right, everybody, look at your study guide.
Wait, she said you look.
What is that for?
Please look at your study guide. Looking at me.
Just put her up.
Looking at me. Thank you. All right, looking at your study guide. You need to know the difference between passive and active transport. That passive requires no energy, active requires energy. You need to know how this energy gets to that pump, that protein pump. How do we power that pump? What's its fuel?
Phosphate.
A phosphate group from,
ATP.
and when we add that phosphate group, what is it called? Phosphorylation. Phosphorylation. Okay. Look at, and you need to know what each of those things, where they occur, and what they transport. Okay? Know that facilitated diffusion is through a protein channel and it's for larger molecules like glucose. Molecules that do not have a charge like glucose. Know how to use the microscope to estimate the size. Okay, here we go. To estimate the size. All right, everybody look. This is my field of view. The field of view is 3 millimeters. Here is my organism. Guys, the easiest way to do this is the following. I divide the field of view halfway, and I know that this is 1.5 millimeters. Is the length of my organism going to be less than 1.5? Yes. So now I'm going to divide my half in half. And now I know that my half of 1.5 is 0.75. Is my organism going to be less than 0.75? What is it? I don't know. You got a choice. Now I don't know what you'll have a choice, so you want the number that is less than 0.75. I mean, the numbers on your test are different, but that's the easiest way to do it. When I was doing this test, that's how I had to do it. All right. Then you know what will happen to animal cells and plant cells in different solutions. Know the meaning of homeostasis and how cells maintain homeostasis by transport. Know what a concentration gradient is and how it works both down and against. When you are moving against a concentration gradient, what kind of transport do you have to have? Active. You gotta have energy. Know how to calculate, know how to interpret diagrams, graphs, and data tables. Calculate. Know your labs. And guys, I don't want you going in there memorizing every word on your labs. I want you to know what happened in those labs and why. Know how to make predictions using data. If I give you this set of data, and if I have an animal cell in this set of solution and it loses water, what's going to happen to the plant cell if I put it in that solution? All right, your free response. You wrote a paper comparing and contrasting prokaryotic and eukaryotic cells. Real quick, what kind of organisms are prokaryotes?
Animal. Oh wait, no.
Prokaryotes are bacteria.
Simple.
If I have a eukaryotic cell as big as a basketball, as big as a I don't know. As big as a basketball, then what size is my prokaryotic going to be? The size of a pinhead. Eukaryotes are big and complex. Prokaryotes are teeny tiny, little, and simple. Read your prokaryote and eukaryote paper. If you did not make well on the paper, read your prokaryote and eukaryote notes. Comparing and contrasting. Any questions? See you tomorrow. We're going to hit a home run.
I'm not going to have it tomorrow. Good work. Good. Guys.
Very
Very good. Who's Zach Morton?